Dr. Naveen Jayaprakash is from the Feinstein Institute. He's going to be speaking about the fascicular organization and fiber composition of the human vagus nerve. The project is part of the phase 2 SPARC program. And so that program is devoted to trying to capture a lot of high quality foundational data sets that relate to the human vagus nerve. And I'm very excited to hear what Naveen has to say on the topic. It's going to be spectacular, I am sure. But I just want to give a plug for not just Naveen, but for the other folks in our project of the Bridging the Body and the Brain seminar series. We have a whole series of speakers that will be presenting all the way through to the beginning of September, at which time we will have learned a whole lot about how bioelectronic medicine, as well as the peripheral nervous system, is so important to health and research related to bridging the body and the brain. We have an exciting announcement that SPARC has been selected by AWS to be part of the Open Data Partnership Program. This is really impactful, especially for projects such as Naveen's, because the sheer amount of data that will be coming from Dr. Naveen's Jayaprakash's project and his cohort and all of the other research scientists involved in the phase 2 of the SPARC program is going to be exceptional. Because of our partnership with AWS, we'll be able to ensure that this data is able to be stored, accessed, and you can move it to another compute node for analysis for your own purposes without charge. So we really think of this as an excellent opportunity for everybody to dive deep into all of this great research and really push the field forward. Naveen's data is going to be on SPARC, but there's also a whole host of other data sets, materials, and other information available on SPARC. And so you can access 2D and 3D connectivity maps. These maps are powered by our knowledge base scan, which allows you to see how all these different populations and areas of the body are interrelated through how the peripheral nervous system connects them all. You can create computational pipelines utilizing the simulation platform OSPARC. This computational platform is free for you to use at this time, and so that allows you to bring in data from the SPARC portal or from other places in order to recombine and do analyses. We encourage you all to contribute to our community. We accept data, devices, and models about the PNS, and we're compliant with the 2023 NIH data sharing mandate. Even if you're not in the United States, our commitment to fair data, and therefore as indicated by the 2023 NIH data sharing mandate, means that we are committed to making sure that your data can be seen, understood in a uniform way, and harmonized with other data sets about diverse topics all together to expand your impact, as well as identify potential further collaborations and stimulate some new ideas. So to submit to SPARC, first off, SPARC is open and fair. We accept individual submissions. So if you have a particular data set that you're working on now and you'd like to make sure others can understand how much work you put into your particular hypothesis and the testing that you did, we offer professional curation. We allow you to reserve a DOI. So if you're preparing that data for a publication, you can reserve the DOI so that the data is curated and is made ready at the same time that you're preparing to publish the manuscript. And so that way, when your manuscript becomes available for everybody to read and understand your interpretation of the data, they can also have a link to go see the data for themselves to get a better understanding of what exactly you're how you came to your conclusions. This builds your body of work because it provides yet another way for people to know how you do your science. The Spark portal will automatically update your ORCID profile. So if you utilize an ORCID profile when you deposit your data, we automatically update that body of work for you so that you can see exactly when that data has been published to the Spark portal. And it allows you to bring your data along with your code and other simulations all in one place. So instead of breaking up your data into different repositories for different aspects of your experiment, the whole experiment can stay together. If you are writing a proposal and would like to plan to share your data on Spark, so multiple experiments at a time in the future, we help with grant writing and language assistance for that data sharing plan. We also, again, provide that professional curation, which is really important for ensuring that the data is harmonized in a way that is understandable by people who weren't intimately involved in creating that data. We are an NIH-approved data repository, as well as a HEAL-compliant data repository. So Naveen's data and all the data from his experiments are going to be in good company. So far, we have over 11,000 samples from 4,000 subjects, utilizing 26 methods in 54 tissues across 7 species. And these are all contributed from over 600 individual contributors. And so there is a whole breadth of information. This whole idea behind Spark is to allow data to be together. All of those subjects and methods and materials across those species are organized into 230. Well, this is a little bit out of date since we are continually publishing new projects. But there's over 237 research data sets, 46 anatomical models, and 46 computational models available on the Spark portal right now. They're 100% available for reuse. And they are used. We have over 12,000 downloads and data sets and models to date, indicating that the appetite for this very clear and robust data sets is real. So you can see along the x-axis here are the individual data sets. So each individual submission is represented along the x-axis, and on the y-axis you can see how how many times they've been downloaded for use by others. And so you can see that whether they're research data sets, anatomical models in the light gray, or computational models in purple, there's a lot of interest in what is contained in these data sets, regardless of when they were submitted and across the spectrum of the types of experiments that are contained on the portal. So with that small introduction to the Spark portal, which is where Naveen's data and his colleagues' data are going to live, I'm happy to introduce Dr. Naveen Jayaprakash. The next seminar, just as a teaser, we'll be going into physiology, looking at the differences in the intrinsic cardiac ganglion neurons between species of mice, pigs, and man. So a multi-species look at a singular, very important neuronal system controlling heart function. So without further ado, I'd like to introduce to you Dr. Naveen Jayaprakash from the Feinstein Institute. Naveen, take it away.
Thank you. Let me share my screen. Can you see my screen in full screen? Sue?
Yep,
Can you hear
everything
me?
looks great. Take it
Oh,
away.
okay.
Take it away.
Just want to say thanks to Sue for organizing the webinar series and also giving me this opportunity to present in this webinar series. Today, I want to talk about the fascicular organization and fiber composition of the human vagus nerve. The project was funded by SPARC. It's a truly multi-institution collaborative effort with some part of the work done in Feinstein Institutes for medical research here at Northwell Health. And the extraction of the cadavers, or extraction of the nerve from the cadavers was done at the Hofstra University, and the microcity work was done at the Temple University. So a bit about a background about the vagus nerve for the audience who don't know what the vagus nerve is. So vagus nerve is the 10th cranial nerve. It's the largest parasympathetic nerve, and it is FDA approved to treat epilepsy and depression, specifically for epilepsy and depression that are drug resistant. And in the clinical setting, they do a whole nerve stimulation. The issue with the whole nerve stimulation is that you do a whole nerve stimulation in a very uncontrollable manner. And we know that vagus nerve, being the largest parasympathetic nerve, innervates multiple organs in the viscera. And doing a whole nerve stimulation is not going to be that efficient. And also, we know that the vagus nerve has both the afferent and the efferent arm. And doing a whole nerve stimulation sometimes might be counterintuitive because the therapy that you tend to do will be inhibited by the other pathways. So to be more specific and also to be more efficient, you need to deliver the stimulus in a very specific manner. So the goal of the project is how can we use the VNS to target function and organ-specific fascicles or fibers in the vagus nerve for more precise therapies. The requirement for that is that we need to understand the anatomical organization. One second. We need to understand the anatomical organization of the nerve at the fascicular level and at the fiber level. So to understand this, first we started with the big vagus nerve. We chose the big vagus nerve because it closely resembles the human vagus nerve in terms of the diameter and the size. And we started to understand how these fascicles are organized in the big vagus nerve using the micro-CT. So what we did was we scanned the vagus nerve with the micro-CT, and when we did the scanning, we were able to see the fascicles inside it. And the fascicles that emerge out of the nodose ganglion are now denoted in the red, and the fascicles that go around the nodose ganglion, we color-coded in green. Now, the fascicles, as they emerge from the nodose ganglion, we were able to track it. Now, as you go down caudally, we can still track individual fascicles, and now you can see that here some of the fascicles are coded in red and some of the fascicles are color coded in green. These are the green is the extra nodose fascicles that go around the nodose ganglion, and the red is the ones that are emerging from the nodose ganglion. You can see that these nodose fascicles from the nodose ganglion and extra nodose ganglion tend to remain separate at the cervical level. And as you go down, they tend to mix. Once they mix, they will give a new color, that is the yellow. And as you go down to the thoracic level, now the nodose ganglion fascicles and the extra nodose ganglions tend to mix completely. So this is now, from this, we can say that there's some level of fascicular organization that's happening at the cervical level. But what about the fibers inside them? So to understand the fiber organization, we used the IHC techniques. I'll explain how we did what kind of staining we did, what are the segmentation protocols we used, and all that when we talk about the human vagus nerve and the staining in that. But in the overall view is that From using these ILC techniques, you can understand the distribution of fiber within the fascicle and also distribution of different fiber types within the fascicle. For example, the myelinated efferent fiber, myelinated afferent fibers, unmyelinated efferent fibers, and unmyelinated afferent fibers. And not alone within the fascicle, you can tend to understand the distribution of these fibers across the nerve too. You can see that the myelinated efferent fibers tend to occupy one region of the nerve, whereas the myelinated afferent fiber tend to occupy the other region of the nerve. This I'm showing it in the left vagus nerve, and the same thing you can see it in the right vagus nerve. Overall, based on this, what you can do is that you can provide a stimulation to the big vagus nerve, and then based upon where the stimulation is provided, you can provoke different physiology response. And the work was published in the Brain Stimulation in 2023, and you can read there if you need more details on it. So the next question that we asked was, does the human vagus nerve exhibit similar fascicular and fiber level organization as observed in the pig vagus nerve? So to do that, to explore this, we started a project focused on the human vagus nerve. The project is called the REVA, or the Reconstructing Vagal Anatomy. It's one of the part of the SPARC phase 2 project by NIH. The overall goal of the project was to dissect nerves from 30 cadavers, 30 left, 30 right, starting from the jugular foramen to all the way to the abdominal branches. Mark and label individual branches as they emerge from the nerve, and perform fascicle level imaging throughout the length of each nerve, which is the micro CT. And after the micro CT, we would want to perform the fiber level imaging, which is the IHC at specific levels of interest or regions of interest. Organize, analyze, and visualize the data, and upload all the raw data and process data to the SPARC data set for the entire community to visualize. So again, the goal of the project was for 60 nerves, left and right, to be extracted from 30 cadavers. Currently, we have extracted 54 nerves from 27 cadavers. And we have done so far 14 females and 13 males. The graph here shows that the sex. The graph on the right shows the age. And you can see here, most of the cadavers are about 80 years old. The extraction of the nerve again was done by Dr. Zinab at Hofstra University, and I'm showing you here an example image in which the nerve was extracted from. This is the region of the nodose ganglion, and this is the cervical level, and you can see the nerve at the thoracic level too. And as we did the extraction, we identified individual branches of the nerve by tracing it to the end organ. Once we trace it to the end organ, we label them and mark them. And I'm just showing you here a zoomed in image showing the nodose ganglion, the cervical level, and the thoracic level. And the image in here in the right shows the extracted nerve with all the branches labeled. So after the extraction is done, the nerve is now subjected to a micro CT pipeline. Basically, what we do is that we segment the nerve into small chunks ranging from 1 centimeter to 5 centimeter in length. And when we immerse this segments into the Lugol solution for it to get the contrast, Lugol solution is going to act as a contrast agent. When you put it in the micro CT, you can see the fascicles inside very clearly. So we did the scanning at 9 voxels per pixel at the resolution where you can see the fascicles very clearly. This is a 2D micro CT image of the nerve, where you can see all the fascicles inside the nerve. And the 2D image is just one image, right? Like we can generate thousands and thousands of images of the 2D and reconstruct the whole segment And I'm showing you here is a 3D micro-CT reconstructed image of the nerve. Now, after that, the same segment is now will get subjected to histology and immuno-IHC techniques, where we take sections at different lengths of the nerve. And I'll tell you the details about this in a minute, but the key idea is that with the IHC, you have the advantage to not only track the fascicles, but you can get to the fiber level and identify different fiber types. I'm just showing you a cross-section image of the IHC image taken at the level of the nodose ganglion. You can see the cell bodies here, and you can see the fascicles that are going around it. Now, to summarize, we can collect data from emerging from two data sets. One is the IHC pipeline, another one is the micro-CT. Both the data set needs to be segmented, and this work was done in collaboration with Theo Zano's lab, where the data gets annotated and segmented using an algorithm that they were able to develop. So this enabled us to track the fascicles and fibers inside the nerve. Now I'm going to talk a little bit more about the fiber level imaging with the IHC. And getting back to that, again, as I said before, after the extraction and the micro-CT, the same segment, same nerve will now be subject to the IHC pipeline. What we did was we collected sections, and IHC we were able to collect only a certain regions where like there are starting above the nodose ganglion and one through the nodose ganglion, and the upper cervical, mid cervical, lower cervical, and whenever there's a branch that happens, we collect at that region too. For example, at the cardiac branch, laryngeal, bronchopulmonary, esophageal, and we take the entire segment. We also mark which side is the anterior, lateral, posterior, ventral, and we paraffin embed them and then take 5-micron thin slices. And once we take the 5-micron thin slices, we stain the nerve with the neurofilament, which stains for all the neurons and fibers, and with the myelin basic protein, which stains all the myelin, and cholinesterase transferase, which stains cholinergic neurons, and then thyroxine hydroxylase that stains dopaminergic and noradrenergic neurons. All the 4 stains were co-stained in a single section, and they were imaged in a fluorescent microscope at 100x resolution, enough to see individual axons inside the fascicles. So based on the expression of different fiber markers, you can determine the fiber type, which indicates the function of the fiber. For example, if it's a myelinated sensory fiber, it's going to be glutamatergic, and it's going to have a function of somatic sensation, mostly from pharynx, larynx, and airways. And if it's unmyelinated sensory fiber, it's going to be again glutamatergic. These are special visceral sensation from all innervated organs. And then you have the myelinated motor fibers. These are cholinergic. These are efferent fibers of the vagus nerve, and they provide parasympathetic input to the innervated organs. Theoretically, it's also possible that preganglionic sympathetic fibers can also be cholinergic. I'm imagining these fibers are going to be, we couldn't differentiate between the parasympathetic and the preganglionic sympathetic, but I'm imagining that the percentage of these preganglionic sympathetic fibers are going to be few in number. Then finally, we have the unmyelinated motor fibers, which are catecholaminergic, which are sympathetic input to the innervated organs. I know even though vagus nerve is a parasympathetic input, we do see some of the fibers, close to 5%, as you will see later on, is that they are TH positive fibers. So why do we care about, what is the big deal with this IHC? And why do we care about this? Because much of it has never been done in the human vagus nerve. never done at this massive scale across the entire nerve and in 60 nerves to deal with. And data needs to be double and triple checked because it will be shared across the entire world. So whatever I'm going to show is just not an example image, but it's not a representative image, but we made sure that each and every image that we take is high quality and good enough to have a resolution that you can determine the different fiber types and the distribution of the fiber types across them. So doing this in 60 nerves was long, painful, and it required a lot of optimization because given that we are now doing it for all the four stains and all the four stains now co-stain and imaged at 100x resolution. So just the breadth of it, the data set that we're generating here is massive. So I'm just going to show you an example image, a cross-section of the vagus nerve taken at the cervical level. And what you can see is that the section was co-stained with neurofilament in green, myelin basic protein in red, cholinesterase transferase in blue, and TH, thyroxine hydroxylase, in yellow, shown here. But overall, when you see here, what you see here is that some of the fascicles do tend to appear bluish in color, and some of the fascicles here down look reddish in color. So let's see why is that. So if I'm going to zoom in the fascicle here, what you'll see here at the one section, you will see some of the fibers are myelinated. You can see the myelin around it. And some of them are unmyelinated here. And within that, you can see there are some largely myelinated fiber and some small myelinated fiber. So if I have to take the neurofilament out, what you'll see here is that some of the fibers, particularly the large myelinated fibers, do express chat. And these fibers, we call it as myelinated motor fibers because they express neurofilament, MBP, chat, but do not express TH. But if I go to a different fascicle around here, the fascicle number 2, and if I zoom in here, what you'll see here is the same. Some of the fibers are myelinated and some of them are unmyelinated. The unmyelinated fibers here in this fascicle do express TH and neurofilament, but they do not express MBP or do not express chat. So those fibers, we're going to call them unmyelinated motor fibers. Now going back to the chat-rich fascicle, what you will see here again is that I'm showing you fibers that are expressing has the myelin and some fibers that don't have myelin and some fibers that are myelinated but they're smaller, but they don't express chat. So the fibers that are neurofilament positive but myelin MBP positive but are chat negative and TH negative, we call that myelinated sensory fibers. And again, in the same panel, what you see here is that in this, you see some of them are unmyelinated and they express only neurofilament, but they don't express MBP, they don't express chat, they don't express TH. So we call these fibers unmyelinated sensory fibers. So far, 4 classifications: myelinated motor fibers, unmyelinated motor fibers, myelinated sensory fibers, and unmyelinated sensory fibers, 4 types. But what I haven't shown you is that lack of expression of motor markers doesn't automatically mean that these fibers are sensory. So we have to still show you that some of these unmyelinated sensory fibers do express sensory markers. So to do that, we used a staining called peripherin. So I'm going to show you another cross-section of the nerve. And in this cross-section, I'm zooming in one of the fascicle. And in that fascicle, we stained that cross-section with the peripherin. And you can see that some of the unmyelinated fibers do express this peripherin marker. And these peripherin positive fibers are surrounded by these remark bundle identified by S100B staining. And you can see that these peripherin positive fibers are also expressing neurofilament. And this is an overlay image here. So with the same fascicle, now I want to know, can ask, if this fascicle do or not. So what we did was we stained the nerve with co-stained the same region with the chat and peripherin. And what you see here is that the fascicle that is rich in peripherin do not express most of the chat fibers in there, but they do have higher quantity of TH fibers. But though TH fibers that are do not co-stain with, do not co-express the peripherin, they are rich in the surrounding region, but they remain separate. Now, if I go to another fascicle here, what you see here is that that fascicle is rich in chat, as I'm showing here, but they're poor in peripherin. And the same fascicle that is rich in chat is now going to be poor in TH. And then I'm showing you here is an overlay image where all of the stains are overlaid on top of each other. Now, from this, what we can say is that just using neurofilament alone, and we can identify all these fiber types. And what we're trying to say is that neurofilament is a good marker to identify different fiber types, and particularly the unmyelinated sensory fibers. Now, what we also show is that neurofilament is an intermediate protein. So we stained the nerve with the PGP 9.5, which is a protein expressed specifically by the neurons. And what we show is that neurofilament co-stains perfectly with PGP 9.5. So just to reiterate that neurofilament can be reliably used as a marker to stain all the fiber types. Now we get into the segmentation and quantification. So to the scale in which we are generating all these data set, it's going to be very hard to efficiently and accurately segment and quantify all these fiber types using a generic software, say like an ImageJ. So which was not that accurate to begin with, and also was not that fast. So we have to develop our own algorithm to segment this data and also quantify all these fiber types inside the nerve. So Shiar Bahadir, an MD, a PhD LMSD scholar, helped us in developing this algorithm, and along with Theo Zano's team members, Lucas and Avantika, we are now able to use the algorithm to segment all the fiber types within the IHC images. So I'm going to show you an example here how the segmentation is done. So I'm showing you a single fascicle in which, as was shown previously, that it was rich in chat. And once we segment it, what it does is it identifies all the fiber types within that fascicle. And if I zoom in, and you can see in the raw image, there's the different fiber types, right? And from the segmentation, it accurately determines what proportion of them are myelinated motor fibers. And now I'm showing you, this is a segmented image, and it's clearly identifying only those fiber types that are neurofilament positive, MDP positive, and chat positive, and other fiber types are neglected. And same here, we can now identify and segment myelinated sensory fibers too here, which don't express the chat, but express neurofilament and MDP. Now, the fourth one would be the unmyelinated sensory fibers, which don't express MBP, chat, and TH. Now, the algorithm was able to detect them and segment them, which are only positive for neurofilament. And finally, we have in another fascicle, which was rich in TH, you can see that the algorithm only segments the data which is TH positive and neurofilament positive. Those will be the unmyelinated motor fibers. Now, based upon the expression of all these markers, we can create a distribution of fiber types within and across the nerve. So from the distribution map, what you can see here is that the myelinated, large myelinated chat positive fibers or myelinated motor fibers are clustered in one side of the nerve, whereas the sensory fibers are mostly distributed all across the nerve. I'm going to show you one more example here where you can see the same phenomenon in another nerve where the myelinated motor is clustered in one region, whereas the sensory fibers are clustered all across the nerve. So from this, what we can understand is that, or learn, is that vagus nerve is mostly sensory, with a very low percentage of this large myelinated motor fibers. Um, but the thing is with the IHC that like you can do this distribution map only a certain region of interest. You cannot really track it in a very efficient manner. So for that, we need to track these fascicles and what is the percentage of sensory component to it, what is the percentage of the motor component to it, and along the course of the nerve. To do that, we have to use a micro CT. So now I'm going to shift gears and talk a little bit about the micro CT. So I'm going to show you an example here of a tissue sample. And in the tissue sample, you can see that this segment is marked with different branches with different sutures, identifying which branch it is. It also marks the region where the foramen, where that's the base of the skull from which all the measurements were done. For example, if I say certain branches coming out at certain level from the nerve, it is based upon the region from the jugular foramen, which is the marking here, which is a loose suture placed here. And the entire segment is now scanned with the micro CT, and you can see the micro CT scanned image here. And from there, we annotate the fascicles inside it, which determines which region of the nerve has the epineurium and which region of the nerve is the fascicles are there. And this work was done by Nicole Carpentier in the lab and Todd Levy and Ty Yari. I just want to give a shout out to the level of work they did. This is a huge work, tracking individual fascicles all the way through the length of the nerve. Gonna move forward. So in the image in the right, the image in the left, I'm showing you the annotated image, starting from the nodose ganglion until the upper cervical and annotated all the way to the mid cervical. From the annotated image, what we did was we tracked the fascicles that emerged from the nodose ganglion in red, you can see here, and the fascicles that go around those nodose ganglion in green. And we call these fascicles that go around the nodose ganglion as extranodose ganglion. And based on that, what we do is that as you track down caudally, the nodose fascicles and the extranodose fascicles, what ends up happening is that the nodose fascicles now will tend to mix with the extranodose fascicles or vice versa. If they mix, those fascicles will give a new color code, as I'm showing it here. That's a yellow. So as you go down caudally, the nodose and extranodose fascicles are now going to mix. And when they completely mix, we give another color, that's the yellow. And you can see that as you go down caudally, most of the fascicles are now mixed. So we can validate that if, for example, if I go back, if I say here at this level, at the upper cervical, this is at the upper cervical level, then this fascicle is mostly fascicles derived from the nodose, and this set of fascicles mostly derived from the extranodose fascicles. Then we can validate that with a micro CT. So in the micro CT, we cannot take in all the region of interest. We can take only in one region of interest. And I'm just showing you at that region of interest, we can compare where the micro CT was done and take the same level and do IHC staining at that same level. And if I zoom in, and what I'm showing you here is two regions. In one of the region where it's close to the nodose ganglion is marked as 1, and another region that is the nodose fascicles that are going around the nodose ganglion. And the fascicle in the region number 1, you can see that there's less chat expression, but they do express neurofilament and an MBP. But if I see in the fascicles that are going around the nodose ganglion, you can see that there's a high level of chat expression. And this is a overlay image showing neurofilament, MBP, and chat. Now, from this, what we can do is that so far I've shown you only a 3-class mixing model, which means it's either going to be a sensory or either going to be a nodose derived fascicles or extra nodose fascicles. And in between is a mixing between the nodose and the extra nodose fascicle, right? Now, what really would be beneficial is that we never taken into account what is the proportion in which the nodose fascicles mixes with extra nodose fascicle. For example, if in this example here, if the parent fascicle is larger and is mixing it's mixing with another parent fascicle, which is mostly motor but in smaller in size, then the daughter fascicle would be mostly of the nodose origin of those fascicles. Now, if the size of both the parent fascicles, say they are similar, for example, the percentage of the nodose and the percentage of the extra nodose fascicle is similar, but the percentage of the extra nodose fascicle contains more of the motor fibers and motor fascicles, then the daughter fascicle is going to have mostly the extra nodose of origin, or will have a different color code, mostly in green in color. So based on that, you can create a graded mixing model, which tell you what is the composition of the fascicle, what is the percentage of that fascicle in relation to the max nodose or max extra nodose. And that's what is shown here in the color code. So if it's red, it's mostly max nodose. Mixed is in the yellow, and in the green is mostly max extra nodose. So with the graded mixing model, I'm showing you the same image here with 3-class mixing model overlaid with the graded mixing model here. And with the graded mixing model, you can actually pinpoint the fascicle and tell exactly what percentage of that fascicle contains the nodose fascicles, or what percentage contains the extra nodose fascicles. So I wish I can zoom in a little bit more. But what you do see here is that 95% of this fascicle is mostly sensory, and only 5% of it has the motor component, or the extra nodose component to it. Whereas as you go down, you see a mixing between the nodose fascicle and the extra nodose fascicles, and now it's close to 87% and 12% in this fascicle, and whereas in the yellow fascicle it's 70% and 30%. And as you go down, it's mostly now it's the yellow fascicle is 50% nodose and 50% extra nodose. So what we see here again is that using the graded mixing model, you can track these fascicles from this upper cervical level all the way to the mid cervical level. And you see that at the mid cervical level, most of the fascicles are now mixed, which means most of the fascicles do have some component of sensory fascicles and sensory or nodose fascicles, derived fascicles in them, and also the extra nodose components of the fibers derived from the extra nodose fascicles. Now, what we did so far is that now we can correlate the finding that we get from the IHC to the finding that we get from the micro CT. In the image in the left, I'm showing you nerve B825, which is taken at the upper cervical level. And you can see here again is that the clustering of the motor fibers in one end and the sensory fibers distributed all across the nerve. And this is done only through the micro CT, not taking into account the IHC here. And this was done using the graded mixing model. And I color coded and also denoted each fascicle here, which is fascicle number 1, also corresponds to fascicle number 1 here. And you can correlate what percentage of IHC motor fibers correlate with nodose or extra nodose fascicles, and what percentage of the IHC myelinated sensory fibers correlated with and then fascicles derived from the nodose. And what you see here is that there's a strong correlation between the IHC myelinated motor and the motor component, which is the extranodous fascicles, and also the IHC myelinated sensory and the percentage of micro CT derived from the nodose fascicles. And what you see here is a negative correlation between the unmyelinated motor fibers and the extranodose fascicles here, and also a negative correlation between the IHC unmyelinated sensory fibers and the microCT nodose derived fascicles. So I'm going to show you one more example here where we did that at the level, at the mid-cervical level. This one I'm showing you at the upper cervical level, and this one at the mid-cervical level. At the mid-cervical level, again, we see a strong correlation between the myelinated motor and the micro-CT derived nodose fascicles here, and also the IHC of the myelinated sensory and the micro-CT nodose derived fascicles. But the negative correlation in the unmyelinated motor and unmyelinated sensory. So this, we are able to do that with the multiple nerves. So right now I'm just showing you example of the 3 nerves that we did at different levels. And again, we see the same trend where there's a correlation with the myelinated motor and negative correlation with the unmyelinated motor and unmyelinated sensory. So far we have done 9 nerves. And out of the 9 nerves, the starting point is going to be the jugular foramen, which is the 0 centimeter. And we can track this separation between these nodose-derived fascicles and extranodose fascicles along the length of the nerve. And what we do see here is that they tend to remain separate at the mid-cervical level. And then they start to mix. The red here shows the minimum point in which this nodose and the extranodose fascicles tend to remain separate. And the green dot here shows the point in which they tend to mix. So far, what we find is that the separation between this nodose fascicles and extranodose fascicles tend to vary between the nerve, between the left and the right, and also tend to vary enormously between the cadavers as well there. But in general, the general idea is that they tend to remain on an average separate between 3 to 6 centimeters and then largely mix around like 7 to 8 centimeters in length, measured from the jugular foramen. So in the clinical setting, what it translates into is that if we move the stimulation electrode a couple of centimeters above, say close to 5 or 6 centimeters, you will have much more efficient way of either selecting a sensory or nodose-derived fascicles or extranodose derived fascicles for the stimulation purpose. With this, I just want to thank the entire lab members. I'm showing you an image here. Nicole Carpenter, who worked on tracking the fascicles and the micro CT data. Parisa here, who did all the staining. Khaled for helping me with the tissue sectioning, and Shiar for developing the segmentation algorithm, and Ibrahim Mugrabi, who has helped me with all the buffers and helped me get the background less with the new buffers that he was able to develop. And Tara Yari is an undergrad who has helped us with the micro CT data. Todd Levy for the micro CT, Zinab for the dissections, and everyone in the Theo Zanos lab who has helped us with the segmentation of the data, and Dr. Mary Barb in Temple University for the micro CT and scanning the nerve. Do you guys have any questions?
Thank you so much, Naveen. I invite everybody to pop your questions into the question window if you'd like to pop those in. I will relay them to Naveen to answer up. And so, Naveen, can you bring us back to the big picture again. So you've got all of this data from, what did you say, 60 cadavers. So how many total nerves are you looking at?
Let me stop sharing my screen and then I can see your face.
Sure.
So
So
yeah.
I'm wondering if you can put it into a perspective of just at the basic level, why is the data why is the experiment so large? Why are you looking at so many nerves um in the left and right? So just a real basic question, but bring us back to that if you wouldn't mind.
So as I said before, we see in the data too, there's a variability from one cadaver to another cadaver, and it could be the sex differences, it could be the demographic differences, it could be the age difference. So there can be multitude of reasons why there could be a difference between whatever we find in one subject or another subject. So it's good to have a larger N to see on an average what is actually happening, right? So that's the reason why it's the 60 cadavers. And we are generating thousands and thousands of images because in each of the nerve, we go ahead and select different regions on an average close to 30 or 35 regions of interest just with the IHC alone. And then again, IHC was done at 100x resolution, and each and individual fibers within the IHC, we are imaging it so that you can see it very clearly. So you can imagine the terabytes and terabytes of data we generate just with IHC alone, and now we add on top of the micro CT, right? The micro CT now allows you to track individual fibers, individual fascicles along the length of the nerve. That again adds a lot of data and all this data again will be published and will be deposited in the Spark portal. So.
Yeah, it looks like it's going to be a real amazing foundational data set, right? You're going to have just a tremendous wealth of information that can be explored and used for hypothesis generation for quite some time. Um, we're getting just a lot of big kudos to you and your group in the question panel right now. Everybody's pretty interested in sort of, okay, they're hungry for more. So, do you have a sense yet on the whether or not there's a relationship between the organs and the fascicular organization in the nerve? Do you see anything there that you're able to speak about whether or not there's a relationship or a correlation?
Correlation between just to understand the question you asked, you're asking me if there is a correlation between the fascicles and what? I did not get that.
The organ in which the vagus nerve is going to be innervating.
Organ specific, I don't have the data yet on that. We have to analyze, for example, we're working on it to say what percentage of this is going into the RLN, and within the RLN, what percentage is the myelinated motor and unmyelinated motor and all the fiber types I talked about. So that's going to be the second work or the second paper that I'm working on right now.
Yeah.
But I don't have a concrete data to present on that right now. That's going to be the next thing that we are working on.
Oh, that's spectacular. And so just for, the non-vagus specialists in the room, which would be me, RLN is recurrent
Recurrent
laryngeal, is that
laryngeal.
right? And what function does the RLN convey? What type of
The
information?
laryngeal, it goes to the larynx.
It goes to the larynx. Okay, so that would
Yeah.
be sensory and motor then,
The
right?
recurrent laryngeal,
Yeah.
right?
Okay.
So it loops back.
Awesome.
So we just not alone do RLN. We are doing right now. We're taking data sets from all the branches to the cardiac, the pulmonary, the esophageal, the RLN, And we're looking into at that branch point, what is a percentage of these different fiber types at the emerging point. That's the next data set we'll try to publish on. I have very preliminary data on that, but nothing concrete to say anything right now.
Right, right. And uh, another particular question that has come up is, all right, so you've done some great immunohistochemistry looking to categorize the different fiber types. Have you looked at any nociceptive type markers like TRPV1 or piezo 1, 2?
That's a good question. I did Trippy one. Unfortunately, it didn't. We couldn't optimize it to work more reliably in here. So we for the unmyelinated sensory fibers, you can further categorize into different types. We haven't done that yet.
Okay. Yep. So another opportunity for future work for sure. Okay, this question comes to us, it's more on the logistics side. Drew would like to know how you store and manage such a large amount of data.
Yeah. So we get good amount of help from the Theo's lab for this. But what it comes down to me swapping the drive, putting a lot of storage within the computer every couple of months, getting the hard disk and storing it locally, and then it gets ported down to a large uh, data storage device as a server that we have within the institute, and all of them gets stored in the server. But
Yeah.
we have two kinds right now. One is a local storage that we have, which is all, in fact, not even the normal hard drives. These are SSDs, so they're more reliable, more robust, more faster. And I have many of them stacked within the computer, and that's how I store them. And then we have this 10G network connection that gets ported into the server room, and all the data now lives in the server, and anybody within our institute can access that data set.
Yeah. And just to add on to that, as this data begins to, be passed on to the Spark portal where it's open, that's one of the main reasons why we're so incredibly excited about the partnership with AWS to make this data open. AWS is going to store the data for free, but they'll also make it available for other people to access, and there'll be no egress fees to access that data. And that means for you too, Naveen, like literally everybody. It's this
Mhm.
this data is going to be something that you'll be able to dive back into for as long as you want, but it also opens it up for other people to really start thinking about their questions. Speaking of, we've got two more, if you don't mind hanging out
No,
and
no.
answering them. Awesome.
Yeah.
Um, Everybody's super excited, along with me. So, all right, this is going back to this idea of the fascicular organization, and we're interested in the sympathetic labeling of the fibers. Do you have any ideas on where those sympathetic fibers that are where are they innovating, where are they coming from? Do you have any idea about that particular little subset that you just snuck in there?
Right. Little early to say I'll dwell a little bit more on the sympathetic side of it, but I can tell you is that the major component is coming from the superior cervical ganglion.
Okay, yep.
And when you do the dissections, what you see is that the SCG, superior cervical ganglion, is like lies along on the side of the nerve, and then they're shooting out these fibers along the length of the nerve. That is one major source of the sympathetic innervation to the vagus nerve.
Mm.
And then when we quantified it, it comes to around close to 5% of the fiber population are these sympathetic fibers. Uh, we still working on that. That's a third phase of the project that we want to work on. So we have different ideas where what the function of this could be and what are the other sources that they are coming from. too early to dwell on right now on that.
Mhm.
But that's something exciting personally for me. I want to work on that. So we're trying to get the Riva project finished within the next couple of months, and we have like maybe four cadavers, or sorry, like yeah, four cadavers left, or three cadavers left to be done. And once that is done, we want to get this data out, published, and then work on the sympathy component.
Yeah. So the SCG, I recall um, Steve Lewis, uh, who was a SPARC researcher as well, focuses on the SCG and the amount of importance that the SCG actually has in the stories that he would tell me and explanations that he would give about the data that he provided, which is available on the SPARC portal. Was fascinating. And so I look forward to be able to look at both of your data sets together to just sort of get a feel for if that's where your sympathetic fibers are coming in, into the vagus. Like, oh, it seems like it would
Yeah,
just be a natural
we
way
yeah
to
yeah
bring that data
we,
together.
yeah
Oh,
we see a huge contribution from the SCG pouring into the vagus nerve, and also we see a difference between to which level these fibers generate that are coming from the SCG and what level they are merging into the vagus nerve. So
right.
all this quantification needs to be done.
Okay. All right. Related, last question, but it's going to be a two-parter. So the question is related to the segmentation tools that you have employed. Can you describe a little bit more about those segmentation tools? And can you remind me, I feel like, um, correct me if I'm wrong, because I probably am, but I recall that in the the demonstrations that you've provided previously, there's like it's not just a straight up and down cable where all the fascicles, whether they merge and split, just go, they maintain their same orientation within the nerve. Is that correct? Do they stay, cable like all the way through? Or I know they split and merge, but is there any torsion or twist or anything like that?
Right. So definitely they're not capable, like they don't maintain the orientation. They do split and merge to huge part. They also change the position in the nerve too, like not alone the side too, all within the nerve too, like within the fascicle too, the fibers themselves
Oh, okay.
rotate.
Oh,
So
very cool.
so that's the kind of work Todd is going to be working on right now to show exactly where these fascicles are, how they are orienting, what is the splitting and emerging events that are happening as it courses along the length of the nerve.
Mhm.
But to answer your question, just they don't stay put in one place.
Yeah,
They move
they don't make
everywhere,
it
and
easy, right?
yeah, and
With
it's
every
with
amount of
it.
variability that could possibly be, it's just there.
Yeah, it's crazy. The amount of movement within the nerve is crazy because even when you're trying to take sections, just few sections apart and try to match it, if this fascicle is this fascicle, maybe did it merge with the other fascicle? So it gets very tricky if you miss certain segments of the nerve and then try to jump and then go to this part and see if you can correlate it. It gets really, really tricky because the movement within the nerve is like, yeah, huge.
And can you remind me about those segmentation tools? That's from Theo's lab, is that right?
Right. It's also within our lab.
Oh, your
We
lab too.
have
Okay, great.
we have an MD Elmazi scholar. His name is Shiar. I'll introduce you to him. He has developed this segmentation algorithm from the ground up using software called Elastic, and he was able to program and train these algorithms to segment these different fiber types. So ours together, we sat down and told the computer like, hey, this is myelinated and this is Chad, segment it this way. And then if this is unmyelinated, segment this has a specific classification. So for each fiber type, we have a classifier, and the classifier was trained in to identify which fiber type it is, and also not to pick the background and only to pick out the real signal. So
I feel like we're just scratching the surface, and I can't imagine how much sweat and midnight hours and everything that your entire lab has poured into this project. 60 specimens, subjects, all of these different modalities, working out high quality and high fidelity techniques that allow you to ensure that the quality is maintained across these differing differing segments as you go through this very large nerve. It is mind-boggling to me. And I'm so thrilled that you guys undertook this project, that you are getting to the point where you can start teasing us with all the information that is going to come out of this to date, and just even the next steps that follow on from there, which is, all right, well, now that we've got this foundation, what does it lead us to next? There's just going to be so much. And I want to thank you for taking time on this day in particular to give your seminar, because you're packing up and you're going to DC for
yeah,
the next couple of days, right?
yes.
What are you presenting at
I'm
and where?
presenting the same work, but I'll be in person at the time. And
Yeah, so
also,
anybody who's interested and in person at the Neural Interfaces Conference in Arlington, Dr. Jayaprakash is going to be there in the flesh. You can ask him all your questions. And I hope that everybody enjoyed this seminar as much as I did. I have too many questions. I can't wait to corner you sometime in the future and just soak in all the information that you have. But thank you very much for taking your time. I can't wait to see where this comes next. So kudos to you and your lab.
thank you. Thank you.
And thanks,
Bye.
everybody. Next, we'll be talking about the mouse, pig, and man, mouse to man, intercardiac neurons. So join us in just 2 short weeks for that. Take care.